Hello, welcome to another video from the conscientious biologist Ben Gallagher. This is the next one in the A-level series, and it's on membranes 2, which is transport through membranes. It's really important that before you watch this one, you've watched the previous one on membrane structure. If you haven't watched that, it's going to be a little bit more difficult to understand this one. So go back and have a look at that one if you haven't done so already. As always, please do subscribe to my YouTube channel if you haven't done so already, and do have a look at all the information that I put on my Facebook and Instagram accounts. Thank you. So it's really important first we do a recap of some of the basic GCSE concepts, because movement through membranes is still by the same principles of diffusion and active transport. So let's have a quick recap of GCSE. I'm going to put a box over here, and down the middle of this box I'm putting a partially permeable or semi-permeable membrane. If I put a load of solute molecules over on the far side, represented by these emojis, you can see that you've got a high concentration over there, you've got a low concentration closer to us. So if we just let diffusion happen, which remember is a passive process caused really by particles repelling each other because they don't like to be all bunched together in concentration, then what we'll see is some of those over there are going to get or will move passively to the lower side. That's just the process of diffusion. Now, of course, really important to remember that the particles won't stop moving. Because particles don't have any intelligence of their own, they won't go from the far side to this side and go, right, whoa, we're at equal levels now. They'll keep moving, okay? So it might be if you had 14 over the far side, they'll quickly move along and you might end up with 7 and 7, they're at equilibrium, but then an extra 1 or 2 might move across, so you're at 9 and 5. So now diffusion reverses and they go that way and you might get to 8 and 7, but now 8 and 6, so one might move back, and they're constantly back and forth. That constant movement trying to maintain equilibrium is called dynamic equilibrium. So diffusion is trying to achieve a dynamic equilibrium. Now, if we're talking about rate of diffusion, the speed of diffusion, it's all dependent on how different the high is from the low. That's what's called the concentration gradient. If one is really high and one is really low, it's like a steep slope. Think if you were on your bike, you'd go fast down a steep slope. If the concentrations are more even, diffusion is slower. So make sure you understand that about diffusion. Let's have a look now at active transport. So active transport is where you want to push them back. You want to go in the opposite direction. You want to go from low to high. Well, to do that, to move against the concentration, you should remember that that requires energy. That energy comes from something called ATP, which is an energy molecule you get from glucose during respiration. But for the time being, just worry about that they require energy, and they require a protein pump in the cell's membrane to push those things through. Now, diffusion might be still trying to bring them back, so your rate of active transport needs to be greater than the rate of diffusion if you're going to get a net movement of particles. That should be very basic GCSE understanding there, but if you don't understand it, please go back to my GCSE playlist. There's one entire playlist on transport through membranes. There's three or four lessons in there. Go back and watch them if any of this is at all confusing to you, because if it is, there's not much point carrying on with this lesson where we're adding A-level detail on top of it. So looking properly at A-level detail of membrane transport. Remember from the last video what a phospholipid looks like, and in a phospholipid you've got the glycerol head at the top, you've got the fatty acid chains at the bottom. The glycerol head is water loving or hydrophilic, the fatty acid chains are water hating or hydrophobic. That's really essential detail. If you don't know what I'm talking about with that, go back and watch the previous video, membranes 1. Now if we shrink that down, we can extend that along to make a line of phospholipids, and of course you have an attached line of phospholipids beneath them to make up your phospholipid bilayer. That's just kind of what lipids do when they're in water, because you had that mycel that extended into this structure. Now, if we're going to start to look at transport through this layer though, and this phospholipid bilayer is essentially what the membrane is made of, then we've got to think about how things move through it. Now, if you've got a high concentration of things on the outside or above this line and a low concentration on the inside, and you've got particles like these little yellow smiling faces, if they're high on the outside and low on the inside, then they might be able to just diffuse directly through the phospholipid bilayer. That's just basic diffusion, but that can only happen if those particles are small and non charged. Okay, if they've got a charge, so a positive charge or a negative charge, they can't just move through the membrane. And if they're big, they'd be too big to push their way through. But things like oxygen and carbon dioxide, they can move through by this mechanism because they're little and they don't carry a charge. Okay, if you've got something big or something charged, then it needs to have a tunnel to go through. Now, those tunnels are built, like most things in biology, out of proteins. Remember, I say it so many times, proteins do everything. If you need a job done in a cell, it's probably going to be done by a protein. So if I put this large protein going right through the phospholipid bilayer now, but still you've got to wonder, how are those particles going to get through it? Well, these proteins have tunnels through the middle of them. Again, this is a cross-sectional diagram. Think of this in 3 dimensions. That protein that I've drawn there is like a tube. It's not flat, so the tube, the hollow, goes through the middle. Now, really importantly with these proteins, they are specific in shape to the particle that they're going to carry. Think about when you were little and you probably had a toy that had different shaped holes in it, like a circle and a triangle and a square, and you had blocks of different shapes, and you had to put the right shape through the right hole. It's exactly that with these proteins. So if those big spiky pink things up there were glucose molecules, then the tube going through the middle of that protein would have to match their shape so that they could go through. They and only they. This is how membranes control what they let in. So if there's a particle that they want to let in, they'll put loads of proteins in the membrane that can specifically carry that molecule. If there's something they don't want to take in, they simply don't put proteins that can carry that particle into their membranes. So by adjusting what protein channels they have in their membranes, a cell can control what it does and doesn't let in and out. Now, hopefully that makes some sense of when you were told over and over again through lower school, when you were told, oh, membranes can control what enters and leaves the cell. Well, this is how, because a membrane can control what or the cell can control what protein channels it has in there. Okay. Now, of course, with this channel, it means those particles can move through. This is called facilitated diffusion. To facilitate something means to help something. So my videos, these are facilitating your learning. We're helping it happen. Well, in this case, diffusion is being helped or facilitated by that protein. So this is facilitated diffusion, and facilitated diffusion is for large or charged particles. So for example, glucose as an example of a large particle, sodium ion, Na+, as an example of a charged particle. Now that's still from high concentration to low concentration though. If we want to move something in the opposite direction, so if we wanted to move something now from down here at low concentration up to the high concentration, this is going to have to be by active transport. Again, that's done by this protein. We've referred to it as a protein pump, but you can refer to it as a carrier protein. What that's going to do, if it's going to move it through in that direction, then it needs to structurally be different to the other proteins. So look at what it's got. It's got almost like a little mouth region sticking up, a bit like the active site on an enzyme. Now what can happen is that particle that's at lower concentration can move into that gap. It could do that just passively by diffusion because there was nothing in that gap. So the particle could move into there by diffusion because there was space. But what happens then is that the protein undergoes what's called a conformational change. It alters its shape, and that altering of shape is what requires energy. So by altering its shape, it can kind of close off at one end and trap that particle inside. That particle now being trapped in that little bit of space is at a high concentration. So when the protein continues to change its shape, that high concentration of particle within can now be opened and it will pop through on the other side. So that's how active transport works, by a protein actually grabbing hold, changing its shape, grabbing on and opening at the other side. It's that mechanism. Okay, that's active transport, and active transport moves against the concentration gradient, but it does require energy from ATP. So those are the 3 main mechanisms through a membrane. So take a screenshot of that if you haven't done so already. Right, let's look at some of the other components that you've got in a membrane now, and we'll link in where those proteins and things come from that we've just talked about with transport through the protein structure. So what we call a whole membrane is the fluid mosaic model. This is because membranes, the lipids within them can move around a bit. They're a little bit fluid. Try and think of it like a ball pit. When you were little, you went to a ball pit, a ball pool, like a swimming pool, but full of balls, and you just dive in, you can push the balls around and push them out of the way. The balls will move around each other. But if you're looking at the surface, it looks pretty solid. That's how you need to imagine the phospholipid bilayer, with those glycerol heads looking pretty solid, but actually they can move around within each other, okay? Within that, you've got a mosaic of proteins, lots of different proteins in lots of places. Hence fluid mosaic model. So if I put up the phospholipid bilayer again, we know that's the phospholipid bilayer, we know that's the phospholipid over there, but even within that, I could label the hydrophobic fatty acid chains and the hydrophilic glycerol head. I'm going to throw in there the protein that we talked about for facilitated diffusion. That's a transmembrane channel protein. That's what you need to label it. Let's put the active transport one in. That's a transmembrane protein pump or carrier protein, and that's for active transport. You can also have proteins that just sit on one side or another though. They're just on the edges, and another word for on the edges is peripheral. So we can label that as a peripheral protein. They can have lots of different functions. Let's not worry about that for now. Okay, so these are the three main types of protein you can have in there. Now, what you can also have are these structures. These are carbohydrate chains, so chains of sugars that can come off from either a protein or a phospholipid. They're attached to them and they can just move off on the outside. There's loads of functions that they can perform. They could be antigens for the cell, they could be receptors. There's lots of different things that those can do. But if you've got a phospholipid or a protein that has an attached carbohydrate chain, then you have to rename it slightly. So I'm going to rename that phospholipid over there as a glycolipid. So if a phospholipid's got a carbohydrate chain attached, it's called a glycolipid. That peripheral protein with its carbohydrate chain attached is now a peripheral glycoprotein. You just put that prefix of glyco onto the word. Okay, so technically on my diagram, that's now a peripheral glycoprotein. Now I'm going to add another thing onto here. The reason why I've just done this as a random green zigzag is because there's no real accepted symbol to use for this. But that little kind of blob, if you see something that's a bit weird drawn onto the diagram, or you see it through maybe a really good electron microscope, photomicrograph, if you see something you go, well, that's clearly not a phospholipid, but it's about the size of a phospholipid and it's in roughly the position that a phospholipid would be in, then it's a cholesterol molecule. Okay, now cholesterol is really, really important within the membrane because cholesterol helps to control membrane fluidity and prevent leaks. Now it does that, if you think back to the last video on structure of lipids, it does this by either donating or taking away hydrogens from the fatty acid chains. That can either make them saturated or unsaturated, and how saturated or how unsaturated they are affects how easily the lipids, the phospholipids, can move around within the membrane. So if you need the cell to not leak and become a bit rigid, you might change the phospholipids so that they're less fluid, don't move around as much, you make the membrane a bit more solid. If you're happy for it to be a bit more fluid, you could change them again using the cholesterol to affect the fluidity and change how the membrane functions and how permeable it is to different things. So cholesterol, another really, really important thing that you get labeled. That's basically it for everything that's in the fluid mosaic model. What I've done though, same as I've done on previous slides, take a screenshot of this if you haven't done already. But like I've done previously, I've given you a blank diagram of it here that you can paste into a Word document, print off, and you can practice labeling all these different parts and write down descriptions of the functions of each one. A really good way to revise this, and the way I've got my class to do it in the past, is to take this diagram and print it or stick it on the middle of a big A3 piece of paper. And then when you're labeling things, you can add all your details. So with the phospholipid, you might want to put a diagram of a triglyceride and explain how a phospholipid is different to a triglyceride. You might want to explain why the glycerol head is hydrophilic, but the tails are hydrophobic. You might want to put a description of how facilitated diffusion works, or add some little diagrams to almost animate in sequence how the active transport protein works. But try and get all your information about membranes down on this sheet. Okay, From here, what I'd like you to do is start looking in more detail at DNA as a biochemical. So head to this video on DNA biochemistry. We look at nucleotide structure and polymerization to make DNA chains. Head there next. If you enjoyed this video, give it a like. As always, please do subscribe to my YouTube channel if you haven't done so already. And on my Facebook and Instagram pages, there's loads of additional interesting bits of biology that will just round out your general knowledge, give you a lot more to talk about on the subject of biology. Thank you.